Okay, so welcome to this next video in the playlist on lipid modification of proteins. Okay, so in this video what we're going to talk about is the GQ pathway, and specifically we're going to look at palmitoylation of one of the members of the GQ pathway, which is the IP3 receptor. Okay, and we're going to look at the enzyme complex which is involved in palmitoylating the IP3 receptor. Okay, right, but initially we're going to just look at the GQ pathway. So, let's start with the GQ pathway. So, the whole story starts with a receptor on the surface of your cell. Okay, and this receptor is going to be a G protein coupled receptor, or a 7-transmembrane receptor. Okay, and it's going to be coupled to a heterotrimeric G protein, and that G protein is going to be a GQ heterotrimeric G protein. Okay, so here, implanted into the phospholipid bilayer of the cell, this is our G protein coupled receptor. And G protein coupled receptors are often abbreviated to GPCRs for short. Okay, so I'm going to try and keep this as unspecific as possible, i.e. as general as possible, that's the word I'm looking for. However, if you do want a specific example of a G protein coupled receptor that is coupled to the GQ pathway, then an archetypal example would be to consider the H1 receptor, and its ligand would be histamine. Okay, right, so you can imagine that our ligand is histamine, and this receptor is the H1 receptor if you want a specific example. But more generally, you'll just have some ligand binding to this G protein coupled receptor. And all G protein coupled receptors have their amino termini extracellularly, and their carboxylic acid terminus is intracellular, okay, and they have these 7 membrane-spanning alpha helices, okay, and this is why G protein coupled receptors are also known as 7TMs, okay, for 7 transmembrane regions. Okay, right, the G protein coupled receptor is going to be coupled to a heterotrimeric G protein. So I'll draw the heterotrimeric G protein here. Now sometimes the heterotrimeric G protein is actually physically linked to the intracellular domain of the G protein coupled receptors. At other times, the heterotrimeric G protein is just anchored in the cytosolic aspect of the plasma membrane, i.e. usually they often have palmitoyl groups that will anchor them in the inner leaflet of the phospholipid bilayer, okay, so they're often attached to the inner leaflet of the phospholipid bilayer, which I'm not actually showing here because I don't really have enough space to put it there, so I'm just putting it underneath here. But the point is, in either case, whether they're actually physically linked to the G protein coupled receptor, or whether they're just in the inner leaflet of the phospholipid bilayer, or at least attached to it, the G protein coupled receptor and the G protein will be close by. Okay, so heterotrimeric G proteins then, they consist of 3 separate subunits which are all different basically. They have an alpha, a beta, and a gamma subunit, okay? And that's why they are called heterotrimeric G proteins. Hetero means different, trimeric means made up of 3 subunits, so they're made up of 3 separate subunits. This one is going to represent the alpha subunit, and this middle one will represent the beta subunit, and this final one will represent the gamma subunit. Now, when you are building a heterotrimeric G protein, you have a huge amount of scope for generating a whole bunch of very different G proteins, okay, because there are 16 possible different alpha subunits that you can use here. There are 5 different beta subunits, and there are 12 different gamma subunits, so you could make a huge number of different heterotrimeric G proteins. Now, we are talking about the GQ pathway, so this heterotrimeric G protein here is going to be a GQ heterotrimeric G protein. So this is going to be GQ. I'm just going to put a bit of colour on here to try and make it look a little bit more interesting. Okay, so I'll outline this heterotrimeric G protein in turquoise here. Now, if the heterotrimeric G protein is GQ, what does that actually mean? Well, basically, heterotrimeric G proteins are named after which alpha subunit you use. They are not named after which beta or gamma subunit you use. So the fact that this is a GQ heterotrimeric G protein, all that tells you is that the alpha subunit is an alpha Q subunit. So remember, there are 16 different alpha subunits. One of these 16 is an alpha Q subunit. So basically, if your heterotrimeric G protein is GQ, all that means is that if you go and have a look at your alpha subunit, it will be an alpha Q subunit rather than any of the other 15. Okay, but it does not, to repeat, it does not tell you what the beta and the gamma subunits are. Okay, so let's say we have our heterotrimeric GQ G protein. Now, heterotrimeric G proteins, like all G proteins, have two states. They have an on state where they are bound to GTP, and they have an off state where they are bound to GDP. Okay, now it's the alpha subunit that has the GTP or the GDP bound to it. Now originally the heterotrimeric G protein is in the off state, so it has guanosine diphosphate bound to the alpha Q subunit. Okay, whoops, here we go. So there's our guanosine diphosphate, GDP, bound to our alpha Q subunit. And when the alpha Q subunit has guanosine diphosphate linked to it, it combines to the beta and the gamma subunit, so the whole thing is assembled up. Okay, now, when the ligand binds to the G protein coupled receptor, what will happen is this G protein coupled receptor will gain catalytic activity. And it's going to turn on the GQ protein, basically. And what it's going to do is it's going to chop off the guanosine diphosphate from the alpha subunit, and it's going to bind a guanosine triphosphate molecule onto the alpha subunit. Okay, so here is a guanosine triphosphate molecule, and this is now going to be bound to this alpha Q subunit here. So this is an alpha Q subunit here. Okay, right, and once the alpha Q subunit has guanosine triphosphate bound to it, it no longer wants to associate with the beta and the gamma subunit, so they break off. Okay, they stay bound to one another, but they're no longer bound to the alpha Q subunit, and this is the on state of the heterotrimeric G protein. So in the off state, all three subunits are bound to each other, and the GDP is bound to the alpha subunit, whereas in the on state the alpha subunit has GTP bound to it, and also it no longer has the beta and the gamma subunit bound to it. Okay, and this combination of beta and gamma is henceforth called the beta-gamma subunit. Okay, right. Now, what's going to happen is this alpha Q GTP that you now have, and it often is referred to as that, it's referred to as alpha Q GTP. This is going to go off, and it's going to activate an enzyme within the membrane. So it will remain attached to the membrane. It might well have some palmitoyl groups that keep it attached to the membrane, and it will move off, and it is going to activate an enzyme which is in the membrane. Okay, so let me show you this enzyme. So there's an enzyme that sits in the phospholipid bilayer of cells, and this enzyme is known as phospholipase C-beta, and for short it's PLC-beta. So P stands for phospho, L stands for lipase, and then the C stands for C. So this is a phospholipase C enzyme, and there are many different phospholipase C enzymes. Okay, so this is specifically phospholipase C beta. So it's the phospholipase C beta enzyme that is activated by the alpha Q GTP subunit. Okay, and once you have activated this phospholipase C beta enzyme, it's going to start working on a substrate which is present within the phospholipid bilayer, and this substrate which is present within the phospholipid bilayer is a molecule known as phosphatidyl inositol 4,5-bisphosphate. Okay, so phosphatidyl and then inositol 4,5-bisphosphate, and for short, phosphatidyl inositol 4,5-bisphosphate is often abbreviated to PIP2. Okay, and let me explain the logic behind this. The first P here is phosphatidyl, Then the next symbol is an I, and that's for the inositol. So Pi is for phosphatidyl inositol. Then this final P is for phosphate. And then we have two phosphate groups off this inositol ring, so it's a PIP2 molecule. Okay, and this is often just called PIP2 or PIP2. Right, so let me try and explain the structure of this molecule to you. Now basically, phosphatidyl inositol 4,5-bisphosphate is just a modified phospholipid, and that's what I hope to convince you of. So, let's now zoom in on the phospholipid bilayer. Okay, so we'll start off with the structure of a normal old boring old phospholipid, and then we'll see how we can modify it to turn it into phosphatidyl inositol 4,5-bisphosphate. And these molecules are a normal component of the phospholipid bilayer. Okay, so let's start off with the structure of a normal old boring old phospholipid. Okay, so here is a little cartoon structure of a normal old boring old phospholipid. Okay, so we'll start with these vertical lines here that I'm now highlighting in orange. Okay, so what do those represent? Those represent long-chain carboxylic acids, okay, which are also called by biochemists fatty acids. So long-chain carboxylic acids is their proper chemist's name, but fatty acids is the old biochemist name for these sort of things. They're basically carboxylic acids with extremely long tails, okay, which are hydrophobic. And basically you have esterified them. You've taken two of them, and you've esterified these two long-chain carboxylic acids to alcohol groups on the backbone molecule of this phospholipid molecule, and that's a glycerol molecule. So this horizontal line in green here, this represents glycerol, okay? And again, glycerol is the biochemist's name for this molecule. The chemist's name for glycerol, the proper name for glycerol, is to call it propane 1,2,3 and then triol. And although propane 1,2,3 triol is a bit of a mouthful, it's a good name for this molecule because it tells you exactly what it is. And propane tells you that this is a 3-carbon alkane, and then 1,2,3 triol tells you that you've got alcohol groups coming off each of the 3 carbons in this alkane, and you've got 1 alcohol group coming off each. Okay, now, to the alcohol group that comes off the first carbon, you have stuck a long-chain carboxylic acid via an ester link. You've done the same to the alcohol group that comes off the second carbon, and then finally to the alcohol group coming off the third carbon, you have stuck a phosphate group, which is here in pink basically, and you've attached that via a phosphoester link, which is very similar to a normal ester link, except that instead of a carboxylic acid group, you've got a phosphate group. Okay, so this is a phosphate group here. Okay, so this molecule then now is a normal old boring old phospholipid, the major thing that makes up the phospholipid bilayer. Now, the other name for a phospholipid, the old name for a phospholipid, is to call it a phosphatidate molecule. Okay, and although you'll never ever hear anyone refer to a phospholipid molecule as a phosphatidate molecule, when people are talking about modified phospholipids, they use this word all the time. This is what this phosphatidyl here in the word, well, in the name, phosphatidyl inositol 4,5-bisphosphate means. It means a normal old boring old phospholipid, then with a bunch of stuff stuck onto it, inositol 4,5-bisphosphate. So to convert this normal old boring old phospholipid into PIP2, we need to now stick onto the phosphatidate molecule, which we're now calling phosphatidyl, just because it's a group. So when you stick a phosphatidate molecule onto something, it's now a group, okay? So the group of a phosphatidate molecule is called phosphatidyl, okay? So it's just like if you've got acetic acid and you stick it onto something, it's then called acetyl, okay? That's what the difference between phosphatidate and phosphatidyl is. Okay, right, so we're going to stick this onto an inositol molecule. Okay, now inositol is going to be stuck onto the other free alcohol group of the inositol ring. Sorry, not of the inositol ring, of the phosphate group. So this phosphate group would have originally had two free alcohol groups. One has been involved in attaching to the alcohol group of the third carbon of the glycerol molecule. The other is now involved to attaching to one of the carbons of this inositol ring. Okay, so inositol then. What is the structure of inositol? Inositol is a six-membered carbon ring, hence me drawing a hexagon, where all of the bonds within the ring are single bonds. Okay, that means that every carbon in this six-membered carbon ring has two free bonds that I still need to fill. So basically, to each carbon, what I do is I bind one hydrogen atom, and then I also stick an alcohol group onto each one. So that means that this ring basically has 6 alcohol groups, one of each one of these carbons, and each one of these carbons also has a hydrogen coming off it. Okay, so that's the structure of inositol here. So this is inositol. So what I've created now is a phosphatidyl inositol molecule, a PI molecule. Now, I don't want phosphatidyl inositol, however. I want phosphatidyl inositol 4,5-bisphosphate. So, what I need to do is stick a few more phosphate groups onto this inositol ring. Okay, so all I need to know now is how am I going to label up the carbons within this inositol ring? Well, basically, I'm going to call this carbon that the phosphatidyl group is stuck onto carbon 1. Then I'll go round, anti-clockwise. So I'll go from 1, 2, 3, 4. Excellent. We're going to stick a phosphate group onto the alcohol group that comes off that 4th carbon of the inositol ring. Okay, and here it is. Right, and then 5 is here. So we're now going to stick a phosphate group coming off that 5th carbon of the inositol ring. Okay, and here it is. Right, okay, so that is phosphatidyl inositol 4,5-bisphosphate. And I hope now that you can appreciate that this is just a phospholipid with this extra little bit added on to its head, basically. So it's just got an extra bit that pokes either into the cytoplasm or into the extracellular fluid. Okay, so it's just a modified phospholipid. And this structure, these phosphatidyl inositol 4,5-bisphosphate molecules, they are a normal component of the phospholipid bilayer. Okay, now, this enzyme, phospholipase C-beta, it's going to act on these phosphatidyl inositol 4,5-bisphosphate molecules. Specifically, what it's going to do is it's going to cut this bond here. So let me show this in this turquoise colour. It's going to cut the bond between the alcohol group that comes off the 3rd carbon of the glycerol molecule and the phosphate group here. Okay, so what is the effect of cutting that bond? What two molecules are you going to end up with? Well, one of these molecules is going to consist of the glycerol molecule bound to 2 long-chain carboxylic acids. Now, these long-chain carboxylic acids are firmly anchored in the phospholipid bilayer, so this molecule basically is going to remain in the phospholipid bilayer, okay? So if I continue this picture of the phospholipid bilayer on, then it's going to continue in here. Okay, so let me just colour it in again. So here are the long-chain carboxylic acids in orange here, and here is the glycerol molecule in green. Okay, and this molecule is what is known as diacyl glycerol. Okay, so this is called diacyl glycerol. Okay, and you can also call diacyl glycerol diglyceride. Okay, so both names are acceptable. Right, okay, now the reason that diacyl glycerol makes sense as a name for this is because firstly you've got glycerol, then you've got 2 fatty acids esterified to it. So where does the acyl come from? So let me explain what an acyl group is. So basically, if you have a long chain carboxylic acid here, okay, so let's say this is our long-chain carboxylic acid. So R just means some arbitrary long hydrophobic group. Okay? Now, when you add this onto a glycerol molecule, you are going to bind it via an ester link to an alcohol group on the glycerol molecule. Okay, and when you do that, you will take off this alcohol group that is on the carboxylic acid group. So effectively, what you are going to actually add on to the glycerol molecule is not the full carboxylic acid molecule, but instead is this structure here, this carbon with an oxygen double bound to it, and then an R group here. And this sort of a structure is known as an acyl group. So that is why this is called a diacyl glycerol, because you have got two acyl groups, which are these two long-chain carboxylic acids, and you have added only the acyl group on. You haven't added the alcohol group of the carboxylic acid group. Okay, so you have effectively stuck on two acyl groups onto glycerol, and that's where the logic in that name comes from. Okay, so that's diacyl glycerol. That's one of these products. Okay, and that has a fascinating role, basically. It goes off and activates protein kinase C, but we're not actually going to be interested in that for the basis of this discussion, because remember, we're interested in palmitoylation of IP3 receptors. Okay, so we're interested in this other molecule here. So we're not going to discuss diacyl glycerol anymore. Okay, we're interested in the calcium signaling. So, the other product then is this inositol ring here, which is this 6-membered carbon ring, which has these 3 phosphate groups coming off it here. Okay, so let me colour this in. So, the inositol ring is this 6-membered ring that I've now coloured in blue here. Okay, and then it's now got these 3 phosphate groups on it. Okay, so what should this what should this molecule be called? Okay, and I'd just like to add, before we move on to this molecule, let me just give you the abbreviation for diacyl glycerol. Diacyl glycerol is often abbreviated to DAG for short. Okay, let's now turn our attention back to this. So, what should this molecule be called? This molecule, well, firstly and foremostly, it is an inositol ring, so it should be called inositol. And now it's got phosphate groups on it, okay? And the phosphate groups are off the 1st, the 4th, and the 5th carbon. So it should be called inositol 1,4,5 trisphosphate. Okay, and for short, inositol 1,4,5 trisphosphate is abbreviated to I for inositol, P for phosphate, and then because we've got 3 phosphates, you put a 3 down there, so it's abbreviated to IP3. Okay, so IP3 will then be released into the cytoplasm. So remember, it was the head that was sticking into the cytoplasm anyway, so it will now be released into the cytoplasm. And it's going to go and act on receptors for IP3, which are within the membrane of the intracellular organelle that is the endoplasmic reticulum. Okay, and we'll continue this discussion in the next video.